Hi, everyone, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today we're talking to Consuelo Walss-Bass. Chello is the professor and John S. Dunn Foundation Distinguished Chair in the Department of Psychiatry and Behavioral Sciences, Director of Psychiatric Genetics Program, Director of UT Health Brain Collection, Co-Director of the Translational Psychiatry Program at the University of Texas McGovern School of Medicine in Houston. Welcome.
Thank you very much for having me.
So Chello studies the genetics and epigenetics of psychiatric disorders to try to reveal the genetic, translational, transcriptional substrates of these disorders in human, both in samples from postmortem tissue and also in cells derived from living humans suffering from them. With us today is Melanie Carliss from UTSA's Department of Neuroscience, Developmental, and Regenerative Biology.
Hi.
And me, I'm your host, Charlie Wilson. So, Chello, the psychiatric diseases are hard for biologists. For most of the diseases, we can't find any structural abnormality of the brain or any consistent genetic signature or any biochemical sign of pathology.
Correct.
Disorders are at least partly inherited, but there's no gene test that could tell me whether I have one of them or not.
Not yet.
Not yet. That's a hopeful way of talking about it.
I think it'll happen, yes.
And for a long time, it's been said there was a strong environmental component of causation, but these have never really been identified in a certain way. And there's never even been a mechanism that was satisfactory to biologists about explaining how environment and genetics interact in the brain.
Correct.
In a mechanistic explanation. So, all of this, it sounds like you're convinced all of this may change, so why don't you offer us some hopeful ideas about what's been learned from genetics and epigenetics about this and that could get us out of this difficult situation?
Okay, yes. So there is a genetic component and an environmental component for sure for all mental health disorders across the board. The genetic component the contribution of the genetic component versus the environmental component really depends on the disorder. So, for example, schizophrenia and bipolar disorder are considered the most highly heritable. That means that the genetic contribution in those two is pretty strong, but the environment is still present and a very important role. And then you go down the board, and for example, for depression, it's about 50% genetic and 50% environmental. So for all disorders, it's really important to understand that both environment and genetics are present, and you really cannot disentangle the two, I think, when you consider the biological mechanisms. What are the underpinnings of these disorders? So the genetic component has been more studied than the environmental one. As far as genetics, I think we are making progress recently. This has happened in recent years with the advent of consortiums. So, for example, the Psychiatric Genetics Consortium. This has been groups of scientists from all over the world. They basically pool samples together of patients from different countries and pulling these samples together and sequencing their genetic makeup. And in that way, we've been able to find what we consider candidate genes. So we cannot call them yet causative genes because we don't know that there are the genes for sure that are causing the disease, but it's pretty credible their association in using these large sample sizes. We've finally been able to identify some. So this is happening for schizophrenia, for bipolar disorder, to a less extent in the other disorders. But the thought is that we will get there. We have to pull more samples, perhaps, and genotype more people. But eventually, I think that the combination of the genes that are causing the different disorders, we're going to be able to have a better understanding of that.
Do we have an idea about the scope of it? I mean, when you say combinations of genes,
yes.
Are you talking about 12 genes or 3 or 1,200?
Very good questions. Actually, hundreds. So for schizophrenia, just to give an example, we're I think now up to 240 or so. I don't know the exact number. I think the latest paper is about to come out soon. But around 240 different genes that have been associated with the illness. It doesn't mean that a particular patient is going to have all the different 240 genes in its particular makeup. No, so different individuals are going to have a different combination of those genes. So the way I think of it is that it's not necessarily, we don't really need to think about the actual gene. What is the gene that is causing the disease? But what are those genes impacting on? What is the network that they're functioning in? What are they impacting? What are they disrupting? That's what I think we need to look at. And that's where the commonality is going to be. The different genes in one patient versus another are going to be impacting the same network.
So as a result, network means biochemical network.
Biochemical network,
So
biochemical pathway.
So as a result, there's some biochemical identifiable defect after all.
Absolutely, yes.
It's just that it's being affected one way in some patient and a different way in some other.
No, I think that the biochemical pathways that's the common thread. So the ones that are coming out in schizophrenia, for example, no real surprise there, but the glutamatergic pathway, the dopaminergic pathway, immune signaling, immune system response, those are altered. Now, there is a big question and a big debate in terms of whether should we start using these clinically to, diagnose or to identify the risk of a particular patient. And that's up for debate. Some people feel strongly that at least for schizophrenia, yes, we can start doing it. Other people feel that no, we're not there yet. And the reason that I actually believe we're not there yet is because we need to study what those genes are doing. How are they impacting the networks? How are they disrupting the cell signaling pathways, the brain system, and then how are they leading to the behavior, which is ultimately what we want to understand. Now, this is where the environment comes in. I think that the understanding now is that, yes, the environment plays an important role, and we know this, just to give you some history, we know this from twin studies. This is the way we figure out heritability of the disorders. Two identical twins in schizophrenia, about 60 to 70% of the time will both of them have the disorder. So what does that mean? That it's not 100% genetic. The environment plays an important role. In the other disorders, for example, in depression, the environment plays a much more important role, right? So we need to understand, how is it that the environment impacts our genome, how the genomes are expressed, are they upregulated, downregulated, and how does that, in combination with the genetic makeup that we inherit, lead then to the disorder. So when I say environment, what do we mean by environment? That's a big question. Everything, basically. Everything that we do, we know now, the way we live, what we eat, whether we exercise or not, how much we sleep, whether we use drugs, whether we have traumatic events happen in our lives, all that is going to impact our epigenetics. So what are epigenetics is epi, meaning the way the chromatin structure is organized, which impacts how our genes are, whether or not they become expressed or not expressed, whether or not they get turned on or off. Let's just put it that way. And they become protein, which is what genes do, right? The environment, everything that we do will impact our epigenome. And that, in combination with the genetic makeup, leads to the disorder in some individuals. And I say it's important because we are all exposed to stress. We all do things. We all have things happen in our lives, but not all of us develop the disorder, right? So how does the environment impact us? And then how does that lead to disorder? That's a really important question. So, I don't know if, Melanie, you have any comments.
And I mean, I think something that's interesting as well is it's not even just what we're doing now or what happened in our childhood. This starts, any exposures in utero will not only affect that fetus, but will also affect their offspring. Absolutely. So we're also thinking about these multi-generational effects of the environment can impact disease too.
Yeah, that is a key point because there's this question about missing heritability. So that's seems like a detective thing, but we know, like I say, that these disorders are genetic and they're heritable, right? But so far, for example, for schizophrenia, we've identified 240 genes. The heritability that they explain is very, very small. So there's this, 5% or something like that?
Yeah, I mean, I think for most complex diseases, the psychiatric disorders are low, but even for some, like it's at most, it's about 20% the effect of genes can be explained.
So some of what we call genetics is really epigenetics. It is the environment of our parents and their parents.
Absolutely.
Yeah.
Yes.
And so I think, a lot of this impact on the heritability, it's related to genetics. I think there's also these pleiotropic effects,
Yes,
these
there's many
interactions
different-
between genes and between
Also rare
epigenetic
variants,
variants.
Again, one other point that needs to be made is that the genes that we've identified so far have been based on what we call genome-wide association studies, which so far have been using SNP arrays that are common SNPs. So we all have single nucleotide polymorphisms in our genome, in our makeup, right? They're called common because many of us have them and nothing happens. So the GWAS study, the genome-wide association studies, are based on those arrays. We're still missing what's called the rare variants, that are not so far being taken into account. So that's another explanation for the missing heritability. So there are several explanations. The pleiotropy, the rare we need to identify more rare variants, and then the inherited the epigenetic inheritance. That's also.
So can we take
important.
the same approach to understanding epigenetics that you take to genetics? You identify the epigenetic modification sites.
Yes.
They are going to be finite in number. You give each one of them a name. You study their relationship with them.
There are different.
Yes and no.
There are. Then we're getting into a lot more complication because the epigenetics, there's different mechanisms of epigenetic modifications. So one is DNA methylation. That is like, you can think of it like a pin on the chromatin. The way the genes are arranged, they're like in a ball, basically, in a big, ball. And the methyl group, I like to think of it kind of like a pin. The methyl groups get attached to these CPG islands on the genome. And if that methyl group is there, if the pin is there, the gene will not get expressed. The transcription factors cannot come in there and bind, and they cannot do their job. If the methyl group is removed, now the gene gets expressed. So that's one way of epigenetic, regulation. But there's millions of CPG islands on our genome.
More than there are genes, or equal, or fewer? More.
Yes.
More than there are
lot
genes.
More.
Well, and it's important also to remember that, there's this common mechanism that Chello is describing of gene regulation by DNA methylation, but then you also have DNA methylation that's not at these sites that is associated with disease, but we don't really know what that does yet.
Yes, exactly. But theoretically, yes.
The thing you were disputing is that I said it was finite.
It kind of is.
Of course, it's finite, but
we can
maybe
measure
it's
these.
Effectively too large to count and to catalog.
No, not necessarily. We are doing it at the methylation level. There's now arrays that we can measure. We can look at the methylation
sequencing.
level at a particular gene or by sequencing. So yes, you can do that. You can look at the methylation level in a disease state compared to a non-disease state, and then see, okay, these genes are more or less methylated compared to the control group. And then they can become markers.
So if we learn some stuff doing that, is that a thing that has helped?
That is something that is happening as we speak.
And a lot of people do that, but I think there's also the added complication that epigenetics itself is variable throughout the lifetime
Because of the environment.
So if you measure this DNA methylation, for example, if you measure it today and measure it tomorrow,
it might be different.
But if it were to cause me to become schizophrenic, then it must be relatively stable.
Yeah, it is.
So some are stable, some are less stable, and that's the idea, is trying to capture what could be stable, but then but also when are these changes important? So we talk about a lot of psychiatric diseases manifest in either late adolescence or early adulthood, but probably they actually start much younger.
Yes.
So you could potentially have these epigenetic changes occurring during childhood, potentially even in utero, that may help to dictate what will happen in early adulthood. But by the time you reach early adulthood, that epigenetic mark may have gone away.
Right, but it had an influence at that particular time in terms of
that
the
sounds
development.
frightening from a discovery point of view.
It does. So it certainly complicates things. Now, let's make it even more complicated. Then you have microRNA regulation, which is another mechanism of epigenetic regulation. That is in itself a whole other world. The microRNAs are these small RNA molecules that bind to an RNA, and they can regulate whether or not the RNA that gets translated into a protein or not. So now there's work trying to identify microRNAs as biomarkers, and there's also thousands of microRNAs. And that's even more complicated because one microRNA can target many different genes, and one particular gene can be targeted by many different microRNAs. So you need to, again, finding the microRNAs that are relevant for a particular disorder, that is also developmentally important. The timing is important. At what period of time did it happen? All that matters. But I think there is hope because more and more, I think now that we are having an understanding of how these things work. That is something that we didn't know just maybe 10, 15 years ago. So now we're understanding it. We have methods to measure them. And the bioinformatic analysis of all these different omics is advancing. So when we do each of these layers, I like to call them the DNA methylation layer, the microRNA level, the transcriptome level, if you can measure all of those in your subjects and not subjects, individuals and people that have the disease compared to people that do not, then that can give you a hint of networks. Again, what we want to do is what are the networks, what are the pathways, the biological pathways that are altered by the combination of all these different levels of analysis. Then that can start to give you ideas of the alterations that are happening.
So it's a huge dimensional problem, but of course one of the things we're getting better at is dimensional problems.
Problems, yes.
Can we collect all of that information about one person?
Yes, we
each
can.
Person's different from each other person, which is a whole other issue.
Yes.
And so that means that one could take modern data analysis techniques and apply them to pull out the pattern of epigenetics and genetics that goes with that.
Yeah.
Is that
Absolutely.
science fiction, or is that something that's happening?
I think it's happening right now.
You can, and there are people that take these approaches to study all of these different levels. But of course, it all costs money. So trying to do that on these large scales to have enough individuals, to really identify, any kind of genetic or epigenetic variant contributing to a disease that's where the difficulty lies.
That is a limitation. But again, I think that now, to me, the answer to that, one of the answers is collaboration. I think collaboration across scientists is extremely important, and we've learned that. We cannot do everything on our own. There's no way that we can one person look at thousands of individuals. So collaborating, making our science known, publishing data sets of these omic levels, making them publicly available so that then another scientist can take that data and combine it with his or her data. And now that's how you grow science, and you eventually come up with the answer, I think, by pulling everything together and collaborating. That's a big thing for me.
That's a sort of, what would I call it, brute force or data-driven approach. But traditionally, people have used a shortcut, a trick. You get enough information about one constellation of things that's a subset of genes or epigenetic factors, and then you think, maybe it's really there, and I could disregard all the others and just bore down into that one.
Yes.
So are there candidates like that? Are there, for anyone, you'd have to, I guess this would be specific for each one of the psychiatric disorders, but is that approach making progress too? Or do we have to know everything before we know anything?
Oh, right. So here's the thing.
I I think there's definitely people that are focused on these candidate genes and understanding what they do, and that's a very important piece of the puzzle. But it's important to remember that even when we have these candidate genes, you might have these, for example, again, going back to schizophrenia, you might have these 240 genetic variants that are associated with the disease. But any one person is going to have a handful of those variants that might be associated with disease for them. Mm-hmm. And a different person will have a different handful of variants mm-hmm. associated with the disease. And so when you find these candidate genes or these genetic variants that you want to understand what is the function, it can be relevant to some people and not others. And how it's relevant is also it's contributing just like probably a fraction of a % towards the disease. So the effect of that variant is going to be very small. So while it helps to understand the function, you need to know collectively, like, how do all of these things function together?
I can see that's true if all of these 240 genes are operating independently. But if they were actually clamped together into groups of 20, and so I could have the disease by having, what would it be, 12?
Probably,
I had 12 of them, one in each of those 20 groups.
Yes.
And then somebody else would have a completely different set of them, but will also have one in each of those 20 groups. Mm-hmm. If it was, of course, I'm just making that up, but if it was like that, then it isn't as bad as it sounds.
No, I think that, yes, each of them is going to have an individually a small effect. But like you say, it's the combination of them, and how do they work together to impact and to contribute to the disease. I think the induced pluripotent stem cell model is actually, in my opinion, very powerful, because that was a limitation that we had before. We could not access the brain of a living individual to be able to study cells, which is what we need to be doing. But now we can take a skin biopsy or a blood sample from a living individual. We know that person's makeup, genetic makeup. We know we will know from genotyping which variants that person has. So let's say it has the 12 of those, 20 of those variants. We can obtain a virtual biopsy, basically, and a virtual brain biopsy by making neurons now from that individual and by making organoids, so brain organoids. And now we can understand how those variants are contributing to disruption of that brain function in that one individual with this particular genetic makeup. And then we can compare it to another individual with a different genetic makeup, but that theoretically, in my opinion, I believe the impact is going to be the same. As I said, the pathways are the same. So what you should be able to see eventually, I think this is what we're going to be able to do, is see that the disruption in the two patients with the two different genetic makeups of the different sets of genes leading to the same outcome. And so that, I think, is going to be the way that we now understand how are the brain systems being altered. And then we can perhaps identify targets for treatment, which is, I think, the goal is, right? We can identify the different combinations of genes, how are they working, and come up with treatments to then perhaps reverse that damage that's being caused. So I think it can happen with the technology now that is available, that making it possible to obtain brain biopsies.
So a side effect of that might be that you would see the defect in the function of a cell that you had derived from some patient, or in the function of an organoid. I don't know exactly how we assess function in organoids, but I'm sure there's a way. And one could just set about to find out what is happening at the cellular level in addition to genetic, epigenetic.
We can look at, for example, let's just say different outcomes, the dendrite extension, and we know that is altered in schizophrenia, right? Or in bipolar disorder, calcium signaling. We can look at those outcomes in our cell model, in our iPSC-derived model. And that is theoretically because of the makeup that person has, the genetic makeup. So
What would a schizophrenic neuron look like? I mean, I don't mean to be glib.
No. Actually, one of the first studies that came out by Christian Bernard in 2011 was they showed that they took schizophrenia fibroblasts from 5 schizophrenia patients, I believe, and 5 controls, made neurons, and they showed that the neurons and the connections between those neurons was very, very diminished in the patient's neurons compared to the controls. And that has something that had been seen at the imaging level and looking at the dendritic spines. So now with the cell model, they were able to kind of validate that. So the idea is that the synapses, the connections between the neurons in a schizophrenia patient, is impaired compared to the control. And this actually comes to the environment and to the reason why a theory is that why is it that we do not see the manifestation of the symptoms until adolescence? If in fact it was a developmental disorder that happened early in life, one theory is because it's during adolescence that we have the process of what's called synaptic pruning. So we're born with a lot more connections between neurons than we actually need. And during adolescence, it's like a tree, those connections get cut, get trimmed, and they go down. That's a natural process. And it happens during adolescence. But imagine if a person with schizophrenia maybe was able to get by before because they had those extra connections, but now when they lose them, the manifestation of symptoms starts to occur. That's a theory, it's a hypothesis, but it's one that kind of makes sense with what we're seeing in terms of the synaptic density impairment. That is something that's a hypothesis and is coming out through the genomic studies, right?
So what are the prospects for treatment? I mean, assuming that we could identify the cellular mechanisms, these are things that could be counteracted somehow, or?
So yes, I believe so, that by identifying the specific targets, the medications that we currently have for schizophrenia, for bipolar disorder, for the other mental health disorders, right now they're not targeting the actual causes of the disease.
They alleviate symptoms.
But it's like giving aspirin when you have a fever. We know the fever is not the cause of the whatever illness you're having, right? It treats the symptom, the fever. That's kind of the same thing that's happening with our mental health medications currently. And that hasn't changed for years. They've made a little bit of modifications to particular medications, and those are the antipsychotics that we currently have. By identifying the actual genes that are involved and the actual mechanisms by which those genes function, the idea is that then now we can develop medications that actually target those genes specifically and help to reverse the problem and help to treat the patients. And there's this, it's sort of used a lot word of, patient,
like the personalized medicine.
Personalized medicine,
yeah.
that's, and, some people say, that's what sort of a catchphrase. But I believe that's going to be the case with schizophrenia. And it happens with cancer. They're doing it already. Cancer, there's different genes that are causing the cancer, and they already are able to target the specific genes in a particular patient to kind of tailor the treatment. I think that's what's going to happen with mental health disorders eventually. We will get there.
I feel
Hopefully.
a lot better already.
I have hope.
So thank you very much.
Thank you. Thank you for having me.
Chello and
Thank you,
Melanie,
for inviting me to be here today.
This has been Neuroscientists Talk Shop.